Welcome to Chapter 10, An Introduction to Cellular Division. We'll discuss division, the cell cycle in which it exists, how that cycle is controlled, what happens when control is lost, and we'll end on a segue of the basics of prokaryotic division, which for them is actually reproduction. Karyokinesis, or mitotic division, is the division of normal body cells. In humans, this refers to any cell with two healthy sets of chromosomes. We refer to these as somatic cells. Having two healthy sets of chromosomes is known as being 2N or diploid in nature. In us, 2N means 46 chromosomes, unless the individual you're discussing has some sort of genetic disorder. This type of division takes us from a single-celled organism, an egg that's just been fertilized by a sperm, to a complete immature adult. What you see here is the initial division in a sea urchin to its 16-cell blastocyte stage onto a fully matured urchin living on the ocean floor. Here's a representation of those 46 chromosomes lined up with their partners in order from largest to smallest. These ordered matched pairs are known as karyotypes. Karyotypes have been created over the years to help us diagnose genetic diseases and see the shapes and structures of the various chromosomes located in the human body. The first 22 pairs are autosomal, and that means that they are not related to the sex of the individual. The 23rd pair are known as the sex chromosomes. In this instance, there are two X sex chromosomes, meaning this karyotype belongs to a female. If there were an X and a Y, this karyotype would belong to a male. The organization of an eukaryotic chromosome and eukaryotic DNA in general is very well studied. The DNA double helix is the most basic form of organization of the eukaryotic chromosome. The double helices are quite long, so we wrap the DNA around histone proteins. Histone proteins exist in bricks of 8 circular proteins that function as a team. The DNA is wrapped around them to keep it from becoming tangled. When you have multiple histone units all together, we refer to those units as nucleosomes. Nucleosomes coil further into chromatin fibers, and those chromatin fibers can coil yet again around a central protein structure. Finally, that centralized protein structure with coiled chromatin is referred to as a chromosome. Duplicated chromosomes exist in pairs to be separated with great care at a later state during mitotic cellular division. What you see here is a sister chromatid. Sister chromatids consist of chromosomes that are attached to one another by their kinetochores, which are made of cohesin proteins. These connecting regions, the kinetochores, are known together as the centromeric region. Mitotic spindle fibers that will be responsible for the movement and the shaken in the system as we undergo mitosis will attach to this centromeric region. You can see the individual microtubules attaching to the kinetochores on the two different sister chromatids. When the sister chromatids are pulled apart, we refer to them individually as singular chromosomes once again. This figure represents a life cycle of a cell. During most of the cell cycle, there is little of that moving and shaking. In fact, 90% of a cell's life cycle is spent in something referred to as interphase. Interphase consists of G1, S, and G2. In G1, or gap 1, there is little physical change that can be seen microscopically. However, the cell is accumulating many of the resources it needs to undergo DNA synthesis. During the DNA synthesis phase, the entire genome is carefully copied and checked for correctness, and that material is packaged with its own histones into its own nucleosome. In the G2 stage, that DNA synthesis process is checked, energy that was repleted during the S phase is built back up, and proteins that will be needed for mitosis are manufactured in mass. The other 10% of the cell's life cycle is known as the mitotic phase. It's split up into mitosis, the active division of the cell's internal components, and cytokinesis, which is the physical division of a single cell into two individual units. So mitosis, or karyokinesis, is a 5-stage process that happens in rapid succession, followed by that 6th-stage, cytokinesis. In prophase, chromosomes begin to condense to the point that they actually become visible with the help of a microscope. Spindle fibers emerge from the centrosomes, and the nuclear envelope begins to break down. The nucleolus is also going to disappear. We're no longer manufacturing RNA, so there's no reason to have a dedicated region. In prometaphase, chromosomes continue to condense and kinetochores appear at their centromeres. The mitotic spindle microtubules will begin attaching to those centromeres, and the centrosomes themselves push away from one another and move towards the opposite poles. In metaphase, mitotic spindle fibers are now completely developed and the centromeres exist at opposite poles of the cell. The chromosomes are lined up at the metaphase plate via a tugging and pulling in between the individual spindle fibers, just like a game of tug-of-war. It's important to note that metaphase plate is not a physical object, but rather just like the equator on the globe is not a physical object, it still has a special demarcation telling us this is the center of the planet. Each of those sister chromatids is going to be attached to a spindle fiber originating from the opposite poles as they line up on that metaphase plate. Step 4 is anaphase. In anaphase, cohesin proteins binding the sister chromatids together are going to start to break down. That kinetochore is going to lose its stickiness. Sister chromatids, now referred to again as chromosomes, are pulled apart in opposite directions towards the opposing poles. Non-kinetochore spindle fibers will lengthen, causing the entire cell to elongate as the chromosomes move apart from one another. In telophase, the chromosomes officially align at their opposite poles and begin to decondense. The nuclear envelope materials will begin to surround each set of the chromosomes yet again to protect that important blueprint information, and those spindle fibers will begin to break down. We don't need them anymore, so there's no reason to keep them around. Finally, we've reached cytokinesis. In animal cells, a cleavage furrow will separate the daughter cells. In plant cells, however, a cell plate will separate the daughter cells. Here's what that looks like in a larger image. In the animal cell, there's a cleavage furrow. There's a contractile ring of proteins that will help pull these two round objects apart from one another by creating a tighter and tighter ring. Think about twisting a balloon to create a balloon animal. As you pinch the ring, as you pinch the balloon, tighter and tighter, you form two separate, very delineated cells. That's what's happening here. In plant cells, however, it's a far more simple process. Much like putting up a barrier in the middle of a very large room, the plant cell elongates and then just builds a new wall down the middle to create two separate spaces. In this instance, it's two separate cells. This is both a cartoon image and a compound microscope image of a whitefish blastula. We can scan the cells of dividing organisms to identify the various mitotic stages. Notice that none of the cells appear to be in the same stage. That's actually by design. If all of these cells were actively dividing at the exact same time, that means that none of them are in G1 or G2, which means that none of them are doing their normal job. While it's important for our cells to divide, it's even more important for them to make sure that they're performing whatever function we need. Otherwise, as maybe your lung was rapidly developing, you wouldn't be taking in any air, and that would be a very bad idea. The length of a cell cycle may vary greatly. The regulation of that cycle might be controlled by external or internal events. External events might include examples like the death of a nearby cell or a lack of a human growth hormone or some sort of missing messenger. If the conditions aren't correct in the environment that the cell lives in, it won't bother to move past G1. For internal regulation, there are three main checkpoints. G1 is looking for overall favorable conditions for cellular division, and it's also considered a restriction point. External influences play very large factors here, and the cell looks for general genomic damage that occurs before that genome has been copied. The G2 checkpoint looks for chromosomal replication errors and ensuring there is no DNA damage. This checkpoint can bar entry into the mitotic phase until the DNA damage is corrected. The M checkpoint occurs at metaphase. It's also known as the spindle checkpoint. In this instance, the cell is ensuring that the mitotic spindle fibers are attached to one half of a sister chromatid to the appropriate kinetochore to make sure that the chromosomes are being pulled to the correct edge of the cell. This is considered by some to be the most important checkpoint because everything from here on out is irreversible. There are many regulator molecules of the cell cycle, and they may act as individuals or work in pairs as teams. Some that focus on positive regulation are known as the cyclins and the cyclin-dependent kinases, or CDKs. They fluctuate at very predictable levels and help to push the cell from one phase or checkpoint into the next. The cyclins work only when bound to their appropriate CDKs, and they must be phosphorylated at specific locations in order to function. CDK and cyclin quantities vary naturally over the life cycle of the cell. When the appropriate CDK and cyclin complex come together, a phosphate-donating protein phosphorylates the unit as a whole in a specific location. The activated CDK cyclin complex then phosphorylates a target protein. That target protein can continue on to advance the cellular cycle. There is a normal fluctuation of these CDK cyclins, and they naturally degrade after they've been used up in their individual phases or to help us get past a particular checkpoint. These are known as positive regulators because their activity pushes the cell cycle forward. There are some negative regulators. RB, or a retinoblastoma protein, is known as a negative regulator. RB regularly exerts control over cell size. When it's dephosphorylated, it binds to a transcription factor that's known as E2F. When bound, transcription is therefore blocked. Genes cannot be read, and proteins cannot be created. However, we can phosphorylate RB, and it lets go of that little E2F molecule, which is then free to bind to the DNA and turn on gene expression, thus advancing the cell cycle. This is known as negative regulation because its normal job is to turn the cell cycle down, or even turn it off. There are two more molecules that are quite popular when we discuss negative regulatory pathways. They are p53 and p21. They're both commonly found in non-functional cells, and actually about 50% of cancers have mutated p53 and p51 proteins. Those numbers I'm saying are just a reference to the molecular mass. p53 is a protein that has 53 kilodaltons as its mass. Normally, their job is to halt cellular processes. p53 directly influences p21, and they help to slow down the cell cycle. If there's any DNA damage, cell cycle abnormalities, or if the cells are existing in a hypoxic state, p53 will step in to allow the cell to attempt some DNA repair so the cell cycle can restart, or if it notices that the damage is just too bad, it will push the cell into apoptosis or program cell death. In the case of apoptosis, all the individual pieces can be recycled and pushed back into the cell later. However, as I previously mentioned, in 50% of cancers, p53 is mutated. So it's not capable of noticing DNA damage, cell cycle abnormalities, or a hypoxic state. The cell cycle continues on, and those cells, with their damage and in dangerous locations, simply become cancerous. These cell cycles are the subject of numerous cancer research articles and even entire textbooks. Let's use that information and apply it to cancer in the cell. Cancer is uncontrolled cellular growth that leads to the accumulation of mutations that leads to more uncontrolled cellular growth. Once you have one mutation, it's easier to get the next and the next and the next, so they can compound rather quickly. There are two major types of genes that are mutated in different cancer cells. Proto-oncogenes consist of a major classification of genes that can be mutated to cause cancer. Proto-oncogenes are genes that regularly act as positive cell cycle regulators. We need signals to push growth forward, but those signals need to be controlled. Mutations may upregulate that positive control, like turning on your CDK cyclin levels constantly. These genes and their products are going to be consistently pushing for growth, and that can cause cancer. On the other side of the coin, there are genes known as tumor suppressors. These genes act as negative cell cycle regulators. They normally turn cell growth off or at least turn it down. RB, which we've just discussed, is an example. Mutating these genes is like cutting the brake lines on the car going downhill. They should be shutting off growth, they should be slowing the process down, but unfortunately they do not. So the cell cycle, while regularly controlled, can get out of whack and cause major illness. Genetic division in prokaryotes is much easier to study and to track. Prokaryotes, including bacteria and archaea, have single circular chromosomes located in a central region known as the nucleoid. Those little chromosomes don't have any histones and therefore don't have any nucleosomes, so it doesn't take nearly as much work to copy their genetic material. They do use the help of some packing proteins that are very close to those cohesin ones we saw in the kinetochores of our eukaryotic chromosomes. Sometimes the chromosomes might have some additional plasmids or little tiny circles that also hang out in that nucleoid region, but it doesn't get terribly advanced. So copying the genetic material doesn't exist in its own separate time frame of the cell life cycle because everything's kind of in one little region. This chart shows you the 5 steps related to binary fission in prokaryote. Replication of the circular prokaryotic chromosome begins at the origin of replication and continues in 2 directions at once. Think about what it looks like when you peel the lid off of the top of a can of yogurt. One spot of initial separation all the way down the middle to the other end. That's step 1 in binary fission. In step 2, the cell begins to elongate and something called Fitz proteins migrate towards the midpoint of the cell. In step 3, the duplicated chromosomes separate and continue to move away from one another towards the opposite ends of the cell. Those Fitz proteins I just mentioned form a circular ring around the periphery of the midpoint between those 2 individual chromosomes. In step 5, the ring directs the formation of something called a septum that divides the cell. The cellular membrane and the cell wall materials accumulate along this septum. In the final step, after the septum is complete, the cell pinches off into 2 separate cells that are known as daughter cells. The Fitzy protein is dispersed throughout the cytoplasm of the new cells and can continue on to help the cell do other things within its cellular life cycle. Thus, the division of prokaryotic and eukaryotic cell ends. Division to create gametes is quite different, and that's what we're going to talk about next.